everyone! This video is a general review of different types of membrane transport processes. There's a lot to cover, so let's jump right in. To start with, membrane transport refers to the movement of materials into or out of a cell. A living cell needs to take in certain things and also send things out of the cell. So a living cell might need to take in food or nutrients or oxygen, and it might need to send things out, such as getting rid of waste products or sending signals to other cells. In either case, this means that something has to cross the cell membrane. And as you learned earlier, the cell membrane or plasma membrane is a flexible layer primarily composed of a phospholipid bilayer, or a double layer of phospholipids. If we were to zoom in on a small section of this cell's membrane, we would see a double layer of phospholipids, probably also some proteins embedded in there, but for now we'll keep it simple. And the properties of these phospholipids lead to certain properties of the membrane itself. For example, the heads of the phospholipids, shown here as these gray circles, are all hydrophilic, whereas the tails of the phospholipid are hydrophobic. And this has ramifications for how things can get across the membrane and what types of things can get across the membrane. There are several types of membrane transport, and they're differentiated by several different factors, including what it is that's crossing the membrane, what kind of molecule or substance, and what are its properties. Also, what direction is the thing moving across the membrane, and how much of it is being transported at a time. If you take a look at this diagram, you can see that membrane transport can be differentiated into passive and active processes depending on energy. So passive processes do not require any additional energy, whereas active processes do require energy. Another piece of information we use when differentiating membrane transport has to do with concentration gradients, because some of these processes are based on and described in terms of concentration gradients. So that's what we're going to look at next. A concentration gradient is a difference in a substance's concentration between two regions. The left-hand side of this diagram shows a concentration gradient where the substance shown in red is highly concentrated in just one region of the area shown. So maybe this is food coloring that's been added to a glass of water. And right now all the food coloring molecules are sort of crowded in one region. They have not spread out yet. In nature, substances tend to move from areas of high concentration to areas of low concentration. And we refer to this movement from high to low as being down their concentration gradient. And this occurs just due to the natural kinetic energy or movement of molecules. It's a passive process. It does not require any additional energy beyond the molecules' movement that they're doing already. Now this process will continue until the substance is evenly distributed or at equilibrium. On the right-hand side of this diagram, you can see that after some time, these particles or the substance is all spread out until it's evenly distributed. So we would say at this point, it is at equilibrium. And the process that happened there is diffusion. Now this can also happen across a membrane. In this diagram, we have a concentration gradient on either side of the membrane. There's a high concentration of this green molecule inside the cell and a lower concentration outside the cell. If this molecule is capable of crossing the membrane, then it's going to move down its concentration gradient from the area of high concentration to the area of low concentration. In this case, that would be from the left to the right. And again, this is a passive process, and it will go until equilibrium, until there's approximately equal concentrations of this molecule on either side of the membrane. Now, there are two types of diffusion across membranes. Some molecules are able to diffuse across membranes simply by moving between the phospholipids. They can just wiggle their way between the molecules. In order to do this, molecules have to be very small and hydrophobic or nonpolar, and they must not be charged. They cannot be ions. Some examples of molecules that can do this would include oxygen gas and carbon dioxide gas. Molecules that do this are said to undergo simple diffusion. So they're just diffusing directly across the membrane by wiggling between those phospholipids. This diagram shows simple diffusion across a membrane over time. As we go from left to right, time is progressing here. So on the left side, at the beginning of the process, we have a strong concentration gradient with a high concentration of this blue molecule on the top side of the membrane. Now, it must be small and hydrophobic and not charged because it is able to just wiggle its way between the phospholipids, moving down its concentration gradient from the area of high concentration to the area of low concentration until, on the right, it is at equilibrium. It's evenly distributed. Now, being at equilibrium doesn't mean that nothing is moving anymore, The molecules can still move in both directions across the membrane. We just no longer see net movement or overall movement in one direction or the other. Rather, they're just moving back and forth with an approximately equal distribution. So that's simple diffusion. But what if a molecule cannot move between the phospholipids? Maybe it's a little bit larger, or maybe it's hydrophilic and polar, or maybe it's charged, maybe it's an ion. Some examples of these would be glucose, which is kind of large, or a sodium ion, which is charged. In order for these molecules to get across the membrane, they have to move through a transport protein. This provides a channel for the molecule to pass through. And these transport proteins are highly specific. Each one allows just one type of molecule to pass across the membrane. So for example, the channel protein that allows glucose to pass would allow only glucose to pass. Sodium could not pass through it. Sodium would need its own specific transport protein. Now this process is known as facilitated diffusion because it's being facilitated or made possible by the protein. In this diagram, we can see a few different examples of facilitated diffusion. On the left, we have potassium ions, and there's a concentration gradient where there's a high concentration inside the cell and a lower concentration outside the cell. So they are moving this way, which is still down their concentration gradient because it's from high concentration to low concentration. This chloride ion in the middle is also moving from high concentration to low concentration down its concentration gradient through a protein. So in all of these cases, we would say this is facilitated diffusion. But what if a cell needs to move a molecule up or against its concentration gradient from low to high concentration? This is going to be a different situation. This means that we're moving the system away from equilibrium. Because this is going against the natural kinetic energy of the molecules, this is going to require energy in the form of ATP, and it also requires a transport protein in order to move those molecules across the membrane. This transport protein acts as a one-way valve to allow things only to move in one direction, and this process is known as active transport, and it's also referred to as pumping. So let's take a closer look at how that special transport protein for active transport would work. It has to act as a one-way valve, which means the shape of the transport protein prevents molecules from moving down their concentration gradient. So if this is my membrane here, and this is the transport protein, if it's going to be active transport, that means that we're going to need to move these molecules from this side to this side. So the first thing that happens is the molecule enters the transport protein. Now usually the transport protein will have a specific affinity and a special binding site for this particular molecule. So in it goes, and then ATP causes the transport protein to change shape. So we get that ATP and the protein shifts to its other shape. So you can see it's sort of switched from being open on top to being open on the bottom. And this allows the transport protein to release that molecule into an area of a higher concentration. So there it goes. And when it's done, the protein goes back to its original shape. So it can go back to doing the same process all over again. So when would a cell do this? When would it need to use this active transport process? There's a few different examples that we see. One would be to take in as much of a valuable substance as it possibly can. For example, when you eat a meal and you break down all of those large food molecules into smaller ones, you don't just want to absorb half of them and have the system go to equilibrium. You want to absorb all of them, get as much energy as possible. So your intestinal cells have the ability to use active transport to take every last usable molecule from your food into your bloodstream. A cell might also use this process to pump out waste. It doesn't want to get rid of just half the waste, it wants to get rid of all of it. So once it's passed, in order to move past equilibrium, it would use active transport to keep pushing the waste product out. And a cell might also use active transport to create or maintain a particular gradient, so to prevent that gradient from reaching equilibrium. Classic example of this is creating a hydrogen ion gradient during cellular respiration in order to power ATP synthesis. So that hydrogen ion gradient has to be constantly maintained in order for ATP production to occur. So all of those involve individual molecules moving across cell membranes. But what if a cell needs to move something larger across its membrane, or if it needs to take in or release large numbers of molecules all at once, rather than just letting them flow through one at a time? For example, what if a cell needs to release really large quantities of neurotransmitter molecules very, very quickly? For these purposes, cells would use what's known as bulk transport processes, and these involve vesicles, which are just small bubbles of membrane that can fuse with or pinch off of the cell membrane. And there are two main types of bulk transport processes. The first one is endocytosis. And endo means into, so this is a process that moves materials into cells. This diagram here shows endocytosis, and you can see that the plasma membrane is pocketing inwards to bring this material, whatever this orange molecule is, into the cell. So by pocketing inwards, it forms a vesicle, and then that vesicle can move into the cell, carrying many of these molecules all at once. It would also work for a large chunk of something that needs to come in and can't fit through a transport protein. The other bulk transport process is pretty much the opposite. It's exocytosis, exo meaning outside. So this is a process that moves materials out of cells. So this diagram on the bottom here shows exocytosis. It starts with a vesicle inside the cell containing a substance that needs to get to the outside of the cell. So this vesicle moves to the membrane and then fuses with the plasma membrane and releases the material to the outside of the cell. In both of these cases, the process works because vesicles and the plasma membrane are both made of the same thing, so they can easily pinch off or fuse with each other. And both of these processes require energy in the form of ATP, and both of them operate independently of concentration gradients. All right, to recap, we went through several different types of membrane transport. We looked at some passive processes, including simple diffusion and facilitated diffusion, and we also looked at some active processes that require energy, including active transport and the bulk transport processes of exocytosis and endocytosis. That's all you need for now. So until next time, take care of yourself, take care of each other.